Yeah, should we start? It's my pleasure to introduce Itamar Ronen, the director of CISC. He's going to talk to us about the MRS method. You know, we had a project proposal presentation a few weeks ago with three new projects doing MRS, and that generated a lot of excitement and interest. So Itamar is going to give us a primer on the use of this method. So over to you, Itamar.
Thank you so much, Chris, and thanks everyone for joining us for this MRS afternoon. I hope one of, well, I shouldn't say many, but at least two maybe, so that we can continue and maybe present a little bit more of advanced concepts in MRS. I really want to apologize that unfortunately I'm flying away tomorrow, so I have to run and catch a cab back home. So after the talk, you will see me just kind of whisking myself away. But please remember that I'm always available for questions, either via email or via visits or any communication method available for you. So please, don't let me be alone in this dreary MRS world. So yeah, today on what's on the agenda, we will kind of give a really crash course in the generation of the magnetic resonance signal, and we will see how we start from one single resonance and we kind of expand it a little bit to include all of those kind of interesting resonances that we are looking at when we are doing in vivo spectroscopy of the brain. We'll talk a little bit about principles of signal acquisition. We'll see that there are a couple of classes of MRS sequences, and I will focus on one class, but I will leave the option for developing a little bit more the topic maybe in a further presentation. And most importantly for you, I think, is how do we process and analyze MRS data with some considerations and caveats that I think it's important to take into consideration when you look at your spectroscopy data. I grayed out the report, how to do MRS reporting. I think it's an important topic. I will also show you a consensus paper about how to report your MRS data. Just put it out there that it is important to at least convey to the reader what is exactly you did and how you did it, because as we'll see, there are many pitfalls and potential points where interpretation might be a little bit off of what you thought it would be. A very fast crash course regarding MR, just to remind those who know it and then maybe introduce it to those who don't. You probably know this graphic representation of an MRI scanner, but what are the components and how do we generate the MR signal? So first of all, first condition is to have a very strong magnet. And the strong magnet generates a strong magnetic field that is typically aligned with the bore of the magnet. You see it here with these three red lines. This is the strong magnetic field that is generated. What does this strong magnetic field do? Well, if we have magnetic material, and we know that magnetic material abounds in nature, but one particular magnetic material of interest to us are the nuclei of hydrogen atoms, the protons. They're actually tiny little magnets. We call them magnetic dipoles. And those magnetic dipoles are oriented randomly in space when there is no magnetic field. But when we switch on the magnetic field is always on, then they align themselves, some parallel, some anti-parallel to the external magnetic field. But overall, what happens is that the sum of all magnetic dipoles of what we have in the sample, which without a magnetic field is zero, is greater than zero when we have the static magnetic field turned on. And this is our best friend in MR. It's called the magnetization. And this tiny little magnetization is the source of the MR signal. Now, in equilibrium, if we don't do anything, this magnetization is aligned with the external magnetic field. But then we take another magnetic field, which is an oscillating magnetic field, radio frequency field, tuned at a very specific frequency called the resonance frequency. frequency or the Larmor frequency. When we apply it to the magnetization, then we are able to tilt the magnetization. Where is my red spot? The tilt the magnetization from being aligned with the external magnetic field to being non-aligned with it, and we tilt it away in such a way that we reach a situation whereby it's, for example, not always, but for example, perpendicular to the external magnetic field. And if we then see what happens to this magnetization, the magnetization starts precessing or revolving around the lines of the external magnetic field as if it were like a top or a gyroscope that revolves around the magnetic field at exactly the same frequency in which we apply the radio frequency field. And this frequency is called omega naught, or the Larmor frequency. And this is the signal that we detect, because when we have a magnetization or a magnet, and this magnet is precessing within a radio frequency coil, then to those of you that are old enough to remember the old days when we had a dynamo on our bicycles, then the dynamo in the bicycle is a magnet that rotates within a coil. And if it is a magnet that rotates within a coil, it generates a current. And this is the current that we eventually detect on the detector that we have from the radio frequency coil. And if we take the evolution in time of this oscillating current, which is at the frequency omega naught, and we do a Fourier transformation, or we represent this oscillation in frequency domain, then we get a kind of a concentration of energy, if you want, or a peak at the frequency omega naught, which is our Larmor frequency. That's the whole story. That's magnetic resonance. We can stop here. And this is your first spectrum of those resonating protons that gives us a frequency, a single frequency that is omega naught. And you see omega naught is proportional to B naught, to the external magnetic field, by a proportionality factor that has this kind of mouthful name, the gyromagnetic ratio. And the gyromagnetic ratio is characteristic nuclei. For example, for the proton, it's quite a high number. For carbon-13, it's a much lower number, about 35% of it, and so on and so forth. But we will deal only with proton resonances. Essentially, this is the most important part of the, let's say, physics that we are going to deal with as an initial part of our story. We will also, at some point, talk about magnetic field gradients. In imaging, the gradients are extremely important. These are those that encode the frequency along the axis and allow us to generate an image. Here, in the context of MRS, well, we will see where it comes. It comes, of course, when we talk about the localization of the signal where we want it to be measured. So, we will not talk about relaxation much, but we have to remember that once the magnetization is away from equilibrium, at some point, the second law of thermodynamics, the simple vector equilibrium, goes back to equilibrium via two different processes. One is an energy exchange for T1, and one is a dephasing of loss of coherence process called T2. Now, let's continue with the story that we had before. So we said that if we have, for example, a bunch of water molecules that are in our scanner, blah, 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 we generate a peak. And the peak is a representation of the resonance frequency, of the precession of the magnetic dipole, magnetic moment of those protons at the magnetic field that we applied at a frequency that we call omega naught. It's perfectly fine. Story is not that simple, because if we have, for example, two types of hydrogen atoms, then we put them in our test tube and we have two peaks, and not just one peak. The question is, why do we have two peaks? And what is exactly the difference, and why should the peak that comes from protons, for example, attached to carbons in a lipid molecule, be different from the resonance frequency of protons that are attached to oxygen in a water molecule? And the answer is simple. We apply an external magnetic field, B naught, and we think that the proton feels a magnetic field, B naught. That's not true, because the protons have a heavy blanket, and this heavy blanket are the electrons that revolve around it, which are also tiny magnets, and they also react to the external magnetic field. And they react to the external magnetic field in such a way that they polarize themselves in the opposite direction, because they have a negative charge. So they polarize themselves in an opposite direction, creating a small induced field that decreases by a little bit the field that the proton feels when it is in the center of the atom. Now, this shielding is expressed in equations that we receive by the electron sigma, and we say that the B induced is the omega, the B naught, the actual external magnetic field, scaled down a little bit by this shielding factor that we call sigma. And the thing is that the shielding factor might differ between different types of protons based on how much this blanket is covering the protons, the electron shield around them. So if you can imagine that if we, for example, are in a water molecule, and the water molecule has an oxygen, and the oxygen atom is what we call a very strongly electronegative atom, which kind of yanks close to him the electrons that are around the proton. Well, the electrons are going to see what's the devious with the oxygen atom, leaving the proton exposed, more exposed, less shielded to the external magnetic field. If it is less shielded, then the local field felt by this proton is higher than the local field felt by the protons that are attached to carbon, which is a less electronegative atom. And this gives us differences in frequency that are tiny, but significant enough to make a chemical distinction between those two types of protons. So how are we expressing it, and what do we see when we are looking at a spectrum that contains more than just one peak? So we saw that we have, in the case, for example, of lipids and water, two different frequencies, and they differ between each other in a certain delta frequency. This is something that because, by the way, historically, people that did use nuclear resonance research, the first ones that identified the strength of it were chemists. Everything has to do with the applications in chemistry. And you see some kind of almost absurd things that they're doing today that still kind of brings back to the days where NMR was in the hand only solely of chemists. So we say that the difference in frequency between those two resonances is called the chemical shift. And the chemical shift is in the order of tens of hertz, hundreds of hertz, thousands of hertz. The resonance frequency, omega naught, is in the hundreds of megahertz. So we're talking about 6 orders of magnitude in difference between those tiny little differences and the basic resonance frequency of those protons. And this is why when we try to express it in like percent or a relative difference, we're talking about not percent, but about parts per million. And this is the reason why we see the ppm scale on NMR or MRS spectrum. Now we have to decide on a reference to say, okay, we are looking at differences, but what is our center of the universe? Where is difference equals 0? Where is our kind of point of origin? We could have decided, for example, that the water resonance would be the point of origin. However, remember the chemists. The chemists looked for something that has the most effective shielding around the protons in the center, so that everything else would be on the right-hand side of it. And this molecule is called tetramethylsilane, and these are the protons that generate the resonance that is arbitrarily defined as chemical shift equals 0 parts per million, 0 ppm. That's our origin, our kind of origin of the system of coordinates of how do we measure from it. And if we express everything as distance, a relative distance from this resonance frequency, the resonance of the lipids is about 1.3 ppm away from the TMS, and the water is about 4.7 ppm. Just to make things a little bit funkier, the axis is also reversed. And the question is, why is the axis reversed? The answer is chemists. No, not only chemists, but chemists in the 1950s, because the experiment at that time was not performed by pulsing the magnetization or applying the radio frequency pulse and then detecting the signal in the reference pole. At that time, everything was kept fixed except for the external field. It was actually B naught that was swept. And it was swept in such a way that this was what we call the area of downfield, and this was the area of upfield. And as you increase the field, those that are more shielded gradually get into resonance. And this is why we flip the axis around. Why we stay that way? Because we're traditionalists. We stick to conventions that have been imposed on us long time ago. But this is the frame of reference, or the way that we present spectra. We present spectra as units in parts per million to remove the dependence on the strength of the external magnetic field. We'd express them going from right to left in terms of ppm. And this is indeed the direction in which the resonance frequency increases. We said this is the less shielded, and this is the more shielded, and this is the highest shielded that we have in terms of proton resonances. Now, moving fast forward to how do we see, or what do we see when we look at the same idea, but not at water and liquids, but when we're looking actually at the brain. And what we do is very simple. We just apply the principles that we saw right now, and we look at the brain. So first thing that we see when we look at the brain is a huge peak that is what is the most concentrated substance in the brain, which is water. Very boring, but not that boring, because that water signal is the reason why we have images. Code it and we get all those nice images. But we said that water is relatively unshielded. TMS is here. Here we have a region of protons, of molecules that are, some of them more shielded, some of them are less shielded. Many of them are detectable with magnetic resonance spectroscopy. And if we take this region, zoom in, and miraculously get rid of this monster peak, by the way, just to give you an idea, that we're talking about concentrations in the body of water of about 40 moles per liter. Twice, because we have two protons, so it's about 80 molars of water protons that we're looking at. Metabolites and neurotransmitters, and all of these are maximum in the millimolar region and typically in the nanomolar and picomolar region. So we are not seeing them at all. Actually, we see a little bit here, this tiny thing we'll see increasing in size once we actually zoom into that. And when we zoom into that region, we saw exactly what we said before, only multiplied by the peaks that are generated by those hydrogen atoms that are attached to different molecules that are in the millimolar level. The level of detectability, NMR is not a sensitive method. It's not, I mean, you know, sensitivity is typically associated with the energy gap that we're looking at. So if there are kind of high energy photons that we're looking at, either in the visible spectrum, etc., or even single photons, here we do need a lot of concentrations because the energy gap that we're detecting is actually very small. So in that respect, we are a low sensitivity method, and we are looking at concentrations that are typically around, let's say, 1 millimolar would be the kind of number for kind of the lowest concentration that we detect. There are factors, of course, that influence it, but that's roughly. But the beauty of those, and you see there are many different substances of which the peaks have been identified and associated with those molecules. And I'm not sure that all of those names mean everything to you, but those names are quite meaningful. So, for example, we do have metabolites that have hydrogen atoms that are characteristics of, let's say, neurons. Most importantly is the strongest peak in the spectrum, which belongs to the CH3 group that you see here of a molecule called N-acetylaspartate. This is what we call a neuronal metabolite or neuronal marker, of which the function has been identified, but we don't really have the time. Glutamate, you do know, it's the kind of major excitatory neurotransmitter in the brain, and it does have also quite a few resonances. This is the chemical representation. We have a CH2 group here, we have a CH2 group here, we have a hydrogen here, and we have all sorts of labile proteins that are not considered to be contributing to the signals. And you can see that glutamate, for example, contributes here, glutamate contributes here. So we have, and also this shoulder level, so contribution of the glutamate signal. We have metabolites that, for example, are more prominent in glia. Which type of glia? Well, it's also kind of a complicated story. There are those specific and those that are even identified as microglial markers, even though no one is really sure. And there is quite a bit of kind of ongoing research in doing metabolomics of exactly how these metabolites are distributed among different cell groups. But overall, we have some sort of an idea of where they are. And for example, the pair creatine phosphocreatine, which is an important pair from the metabolic standpoints and the oxidative phosphorylation standpoint. These metabolites are essentially relatively equally distributed among cell types, give rise to the peak at 3 ppm and the peak at 3.9 ppm. Again, we have a CH3 here and a CH3. If we look at the fuller representation of what we call the neurochemical profile, we can identify all the way up to 18 or 19 metabolites, so-called, that we can identify with magnetic resonance spectroscopy. This is just, again, to give you a little bit of an idea of what we're talking about. There are neurotransmitters, and there are osmolytes, and there are, for example, glutathione is quite interesting from the neuroprotection standpoint. So we have different metabolites that we can identify with different certainty of their identification, and all of them contribute eventually to this beautiful spectrum that you see here. But just to remember a couple of things, each metabolite has a distinct contribution. Well, some metabolites have overlaps, and one of the overlaps is here. For example, creatine and phosphocreatine have an almost identical spectrum. They're really not identifiable, which is a shame, because if we could have identified them properly, then we could have tracked the exchange between creatine and phosphocreatine, mediated by the creatine kinase cycle, which is an important metabolic cycle in energy delivery in the cell. We can do it with phosphorus NMR, but we're not talking about phosphorus NMR here. Another interesting overlap that is quite annoying to many people around here that are interested in GABA is between this peak of the GABA and this annoying peak of what we call the macromolecular baseline. Macromolecular baseline is a kind of empirical evidence of the contribution of macromolecules that are big enough to give broad lines and small enough to still tumble and give us a signal that survives enough so we can actually detect it. And you see, this is an experimentally acquired macromolecular baseline that is really not interpretable, but has lots of nuisances that interfere with quantification that we will talk about later on. And indeed, there is a significant contribution from macromolecules. A very quick run through, I just want to make sure that I'm good with time. A very quick run through some applications of MRS. One of the major applications is in cancer. What I'm going to show you here is a relatively ancient application, kind of a characterization of standard spectra these days. Actually, MRS is one of the unique tools used for actual diagnosis and follow-up and treatment of a very specific metabolite that we did not mention here. It's 2-hydroxyglutarate, kind of an important metabolite in phenotyping a specific type of tumors. But in general, you can see that if we're looking at just standard parietal white matter versus an anaplastic astrocytoma versus the more severe case of glioblastoma multiforme, you see that there are qualitative differences that can readily identify on the spectrum. For example, the N-acetyl aspartate, which we said is a neuronal marker, goes down significantly when we go to the tumor process. On the other hand, choline, which is a marker for membrane turnover, is actually increasing quite dramatically. In this case here, there is also an evidence of astrocytosis by the increase in myeloinositol signal, and here everything simply goes down because tissue is completely destroyed. So we have observations that are related to what we see in different types of tumors. In the case of demyelinating metabolic disease, again, this is an excellent adrenal leukodystrophy. It is genetically transmitted demyelination disease that affects young children. And in this case, you can see that in the articulated image, where you can see the deficit kind of going from the occipital regions towards the frontal regions, already at the stage where the contrast is not visible in the contrast in the frontal region, there is already a significant decrease in NAA, a significant increase in choline, and also in bioinositors. It's quite vividly visible here. This is a strange case of the disease where there is creatine deficiency. This is the spectrum taken from this subject that has no creatine peak, which is quite curious, but then supplemented with creatine, the creatine peaks awaken to life following the treatment. I will skip the Pompe disease. This is actually a study that I'm involved in Leiden, where we are looking at deposits of glycogen in children in the brain. Glycogen is typically something that you do not detect with MRS, because the levels that are in tissue are so low that you don't consider it as a contributor. But in glycogen storage disease, you actually can detect glycogen. Quickly about Alzheimer's, again, volume on the posterior cingulate can detect an increase in glial metabolites, a decrease in neuronal metabolites, and you take the ratio of those two, this kind of trend becomes more statistically significant and more visible. And this is really to run very quickly through, and just to kind of finish with a bang related to functional studies. This is one of the most beautiful functional MRS studies performed by Sylvia Manja and her colleagues in the University of Minnesota and the CMR. And this is essentially doing a functional study where we are not looking at changes during time, but actually at metabolites during time. And this is, for example, the changes observed in glutamate signal and the changes observed in lactate signal as a function of visual stimulation. These are stimulation periods and rest periods, and you can see that the time course indicates that there is an increase in both. There is a decrease, by the way, in glucose and in aspartate, and this is all according to what we expect to see in terms of glucose metabolism in that particular region following activation. That's, I think, my last word about application, just to get things kind of started with respect to what can we do with this. And I put a little summary, kind of telling us about the chemical shift and how the signal is generated, and about the uniqueness of the contribution of each metabolite to the MR spectrum. There are additional features to the spectrum, mostly the interactions between sphincters themselves that I have not put in here. That's for a more advanced talk, but to remember that the spectrum is not just chemical shifts. We talked about the level of predictability that we should always keep in mind when we plan an experiment. What is that we expect to see? We're not looking for serotonin, we're not looking for dopamine, we can look for glutamate, we can, with an effort, look for GABA, for example. Okay, so this is to be remembered, and again, the fact that we have a kind of a differential distribution makes them a nice metabolite, a nice cell type markers that is modulated by disease processes or even by just normal physiology. Now about the methods. Generally speaking, we divide magnetic resonance spectroscopy into two classes of methods. One is single volume spectroscopy, in which we localize the signal in a box and detect what's in it. And one is spatially resolved spectroscopy or magnetic resonance spectroscopic imaging, in which we essentially do something that is very similar to imaging, only the content of each voxel is a little spectrum. So we can actually look at a bigger field of view and encode in such a way that each box here is represented by a spectrum. We can then quantify the metabolite of interest and see how it is distributed in a more kind of broad region. There are advantages and disadvantages of each method. Depends on what you want to look at, what you want to measure, and both methods are, of course, available at CISC, so it's something to be planned if indeed it is of interest. So it really depends on trade-offs. The single volume gives a higher spectral content, it is shorter to acquire, it is not spatially resolved. Spectroscopic imaging, larger coverage, but lower spectral content is, of course, takes longer to acquire. And the principles are quite overlapping. So if we talk about single volume MRS, many of the principles, of course, will be relevant also to spectroscopic imaging. How do we actually localize the spectrum? So far, we said we just excite and we get a signal, but we want to get it from a specific region. Here come to the head, magnetic gradients. Magnetic gradients are applied in a very nice way that is essentially very similar, identical to the way that we select a slice in imaging. How do we select a slice in imaging? Crash course in slice selection. In imaging, if we put the subject in the scanner and the field is completely homogeneous, then the resonance frequency of the protons is completely homogeneous. And if we apply our radio frequency pulse, it will not care whether we are in the feet or in the head. Everything is going to be excited. Of course, the coil is big enough for so region, and we have no ability to select the slice for observation. The selection of the slice depends on our ability to generate a gradually changing magnetic field along an axis of interest. And this gradually changing field is called the magnetic field gradient. The gradient essentially is a linear change in the strength of the magnetic field, for example, along the length of the body. If the magnetic field changes, then the resonance frequency changes. And if the resonance frequency changes, then if we apply the radio frequency at a resonance frequency that is fit with the magnetic field in the chest, then we select a slice in the chest. If we increase the frequency to match with the magnetic field here, which is slightly higher, we move the slice to that region. So that's the basic principle of application of a radio frequency pulse simultaneously with the magnetic field gradient allows us to select the slice. Without getting too much into detail, the application of radio frequency pulse does not excite a single frequency, but excites a bandwidth. And this bandwidth is dictated by the shape of the radio frequency pulse. And the bandwidth allows us to modulate the thickness of the slice. So, for example, narrow bandwidth pulses will excite a thinner slice for a given gradient. But we can also modulate the slice thickness by changing the slope of the gradient. All of this is summarized in this link here between the bandwidth, the slice thickness, the gradient amplitude, and our very good friend, gyromagnetic amplitude. Now, if this idea is understood, then we can go on and understand the simplest of localization sequences, and this is the point result spectroscopy, or TRES. We start with a slice selection, and this is the excitation pulse coupled with the gradient. This is the signal that is generated by it. It looks like this because this is the signal that decays in time. This is the time domain signal we generate with this pulse, and we comply with the idea that we apply the gradient, apply the radio frequency pulse, and thus selected, for example, this slice with the gradient operating in this direction. Okay, third of the job is done. Next stage is to apply another pulse. Now, this pulse is not a 90-degree pulse, it's an 80-degree pulse. Again, this is MR physics. Some of you may remember that this sequence where there is a 90-degree pulse followed by a 180-degree pulse is a spin echo sequence. Spin echo sequence will generate an echo of the signal that we excited to begin with our first pulse at a given time given by the distance between those two pulses. It's a miracle discovered in the 1950s by a physicist called Erwin Hahn, called the Hahn spin echo. And the echo that is generated here is an echo that is generated by spins that felt this pulse and this pulse. But this pulse excited spins in a slab that is perpendicular to the first slab. We selected another direction for the gradient, meaning that the only spins that will generate this echo will be along this sausage that is round or kind of grayed out here. Can you imagine what comes next? Next is another 180-degree pulse with another spin echo. And the spin echo here, and the 180-degree pulse here, is coupled with a gradient in the third direction. So we have three perpendicular directions, three pulses, each of one selects or excites or refocuses the magnetization, and only the spins that felt all three pulses, meaning in the intersection among all these three slabs, will eventually give us the signal. That's the simplest localization sequence. We acquire the signal here. We have now another parameter to consider. This is the echo time of the entire sequence. Typically for PRESS, the minimum echo time in clinical scanners would be around 20 milliseconds. We can go all the way up to very long echo time, but then we risk losing the signal because the signal, of course, dies relaxed. So we have kind of a sweet spot and a choice to make which echo time do we use for our sequence. Okay, we have lots of sequences, press, steam, laser, and semi-laser, all fancy acronyms. We are totally obsessed with acronyms. All of those sequences that I mentioned here are based on the three pulse slab selections. Press, steam, semi-laser, laser, all exactly the same principle with different kind of implementation. Other possibilities exist, we will not focus on them right now. Now, how do you choose your sequence and your parameters? Let's start with the parameters, and the one parameter that we're looking at that we learned from the previous slide is the echo time. We realize that when we have a very short echo time, then we catch all of those signals that have not decayed had we looked if we applied a longer echo. And this is, for example, these are two examples of signals from parietal white matter, occipital gray matter, at short echo time and at long echo time. And you can see that there is quite a significant difference in the intensity of the signal, and these are scaling properly to one another, but also in the complexity of the spectrum that comes from each case. So it really depends on what you want. Short echo time is better SNR and get more metabolites. Longer echo time, it's a simpler quantification. The molecular signal is gone away. Many of those so-called scalarly coupled systems are also gone away. And if you're only interested in a very simple set of metabolites, go for long echo time, why not? You don't really use it all. T2 of metabolites is quite long. There is a loss in signal, but you can gain some benefits by using long echo time. One of the major benefits of using long echo time is actually the detectability of lactate. Lactate has a relatively long T2. Lipids and macromolecules that overlap with the lactate have a relatively short T2. You go for a long echo time, signal from the macromolecules goes away, and the detectability of lactate, which, by the way, in the normal brain, very, very low, even though Cynthia Mancha showed this functional MRS with the normal brain lactate. But this is actually a metabolic syndrome, or Leigh syndrome. And you can see that in this metabolic deficit, there is a very significant peak of lactate actually. It's a doubling that is detectable, and we are sure that it is lactate and not macromolecules and lipids because we eliminate them by using a long echo time. How do you choose a pulse sequence? Well, that's a very long story and a very long debate. Put 5 MRS scientists in a room, have them fight with each other. What's the best sequence? Not for us to choose now. There is the accuracy of localization, sometimes metabolites of interest, sometimes specific absorption rates is an issue. So, for example, semi-phase is a very good sequence. It is RF intensive. So we go to high field, can be quite a restricting thing with respect to the repetition patterns. So there are different considerations. We will not get into the number, but just to give you an idea that there are issues that are involved and that we are trying to kind of come up with recommendations that will help people make the decisions of what would be the best way to choose. Now, you start measuring. It's very important to optimize the measurement before actually you start measuring. I want to focus on two variables that we optimize prior to the acquisition of the signal. One is what we call V0, V naught shimmy, and one is water suppression. So that these are really two dominant factors in spectral quality. Let's start with the homogeneity. The homogeneity of the spectrum, the homogeneity of the external magnetic field is something that we very much care about, because if we have an inhomogeneous magnetic field, the resonance we're looking at are broadened by the fact that we introduce a variance and variability by having a distribution of the magnetic field within the area that we're looking at, and not a very sharp, well-defined magnetic field. And indeed, the spectral lines that are associated with the spectral acquisition at a very well-shimmed magnetic field and a not so well-shimmed magnetic field, well, you can see the lines become broader, but the spectral content is the same, so something has to give. So the amplitude goes lower. If it goes lower, you will lose signal to noise ratio, you lost the signal. So it's very important to have very good shim. 10 hertz for line width at 7 Tesla is very good. 10 hertz for a line width at 3 Tesla is not so good. We actually want to get 3 Tesla at the level of about 4 to 6 hertz line. So it's important to make sure that you do quality control and make sure that the line width is what you expect it to be. Otherwise, you just lost signal for free because you could have actually do it and do it better. Having said that, there are regions in the brain where it's more difficult to shim and those that are simpler to shim. Again, all things that come with experience, we will see that that not everything is very well defined and homogeneous. But just give you kind of a sense of the thing. So bad shimming results in lower SNR and loss of spectral resolution. We don't want that to happen. Water suppression. If you don't suppress the water, you don't suppress the solvent, you end up with peaks that end up on the shoulder of this huge peak that is the source of the water. The quantification becomes so not so good. Also, if we have two signals that are very, very different in intensities, our receiver gain should be accommodated to have all of this signal, but then we end up undersampling the signal coming from the small metabolite. So that's also important. Water suppression model is an actual part of the pulse sequence regarding shimming, not something we do besides the pulse sequence. But the water suppression is a model that we attach to the pulse sequence. And bad water suppression results in distorted baseline, improper sampling of the weak metabolite signal. Last word on acquisition, things that should be relevant to us is the number of averages and the voxel size. This is just to give you an idea of what happens when we increase the number of averages. Statistically speaking, the signal will just add to itself, so will increase as the function of the number of averages. The noise is the stochastic entity that will also increase, but as a square root of the number of averages. So overall, the increase in SNR is related to the square root of number of acquisitions, which means that from some point on, you start really kind of paying a very dear price for averaging. You have 64 averages that take 6 minutes, that take 5 minutes. You want to double it to 128, thinking that you gained a lot. Well, you didn't gain, you gained something, but not a lot. You gained about square root of 2, which is 40%, which is not too bad. But one has to consider that the position longer, things start moving. If things start moving, kind of limit the amount of improvement that you can do. Unless you process the data correctly, which we will get in a second, if we still have time. Voxel size is very, very intuitive. You increase your voxel, the noise is independent of the size of the voxel, the signal is dependent. You increase the voxel twice, you get twice more signal. So in order of trade-offs, one has to consider between number of averages and voxel size, because you increase your voxel size, well, you get more signal, but also get less specific to the region that you want to actually send. It's quite a bit of trade-off that one can do if you kind of think about both of them as you think about an SNR target, and you kind of optimize both voxel size and number of averages to reach your goal in terms of SNR. So just to summarize very quickly this part, localization, we have two ways. We have a plethora of single volume memory sequences with different characteristics, and the choice of echo time is really based on the question asked, and mostly related to what we're interested in. But it's very important to prepare the acquisition properly. That's our role for giving you high quality spectrum. And SNR will depend on various variables that we just mentioned in the last slide. My last topic of the day is about processing and analysis of NRS data. To distinguish between the two, processing is everything that has been done to the data once it leaves the scanner before you go to the quantification, and then we analyze the spectral, which includes the most important part, which is the estimation of the how much metabolites we have each time, or quantification of the spectrum. But we also have, of course, other topics that come out of it, quality control, and statistical analysis, and so on and so forth. I want to focus on these two in the processing and these two in the analysis. So first with the processing, and I promise that every term is cryptic will hopefully become clearer as we talk. One of the things that corrupts spectra is the main thing that makes the localization happen, which is the application of the magnetic field gradients. We apply magnetic field gradients, we switch them on and off within the realm of an existing static magnetic field. The static magnetic field doesn't like this switching on and off. We switch magnetic fields on and off. It creates fluctuations in the magnetic field, and fluctuations in the magnetic field end up in kind of making a concertina of our evolution of the signal, because fluctuations in the magnetic field, this fluctuation is the resonance frequency. And if the resonance frequency goes during the acquisition, we end up with a corrupted spectrum. And this is an exaggerated view of corrupted spectrum that contains what we call eddy currents. So eddy currents are those kind of spurious currents or spurious fluctuations of the magnetic field that are caused by the switching on and off of the magnetic field. But luckily, we can take care of eddy currents for the most part. What do we do? We acquire the signal of the water alone. If we acquire the signal without water suppression of the water alone, with the eddy currents that come with it, it's very easy to characterize these fluctuations of the magnetic field, because we just have a single peak to consider. And these fluctuations essentially are expressed in the change of phase of the signal along its line. Once you have this phase, with a simple file, you can divide your spectral data by this phase vector and clean your spectrum from the eddy currents and get a beautiful looking spectrum as it appears now. What is the requirement? The requirement is that we acquire a very short acquisition of the water signal, non-water suppressed signal, before or after we do the experiment. This is typically embedded within the acquisition of the signal. It's a box that we check. So we make sure that we have a water reference which to do the eddy current correction. Spectral registration is another thing. We talked about the drifts in the spectrum that happens over time. It's not the average, we require several acquisitions of the same spectrum. But if we look, for example, what happens to the frequency during, let's say, 128 acquisitions, then we have a frequency drift here in the range of 7 Hertz. Lucky it is 7 Hertz. Sometimes it can be more than that. If you had a functional MRI experiment before that heated the gradients quite a bit, then the drifts can be about 100 Hertz. So it can be quite significant, or it can be very benign, but you have to make sure that you can correct for that. This is the same thing when you look at the phase of the signal. So this is a more of a jittery, this is more of a trending kind of thing, and both of them vary with the experiment. And you can see when you look at those stacked spectra one after the other, you can see that they are not totally aligned to one another. And then we perform in a similar way that in imaging you do image registration. Here we do a spectral registration. We align all of those spectra by fine-tuning the frequency and the phase so that they are aligned to one another. And the result can give you a boost of about 30% in SNR without having paid any price. You didn't acquire anything else, it just means the data that you have, but it is very important to save the individual acquisitions, not to average them online. And this is something that now we do routinely, because this is part of the post-processing that we do when we acquire them. Quantification, and that will be the last topic that I will talk to you today. We already alluded to the fact that the spectrum, the resulting spectrum, the measured spectrum, is essentially a superposition of all of the individual spectral contributions from the various metabolites that we measure. So if we have a priori knowledge of how the spectra of each metabolite looks like and the conditions of their acquisition, same sequence, the same epoch time, the same everything, then we can come up with a model. And the model is that the spectrum, as we measure, will be a fitted spectrum plus some sort of a residual, some noisy residuals. And the fitted spectrum will be the sum of all the individual spectra of the individual metabolites, which you can obtain either by phantom, scanning them one after the other. N-acetyl-aspartate, prepare phantom scan. Prepare phantom scan. Not anymore. No one does it anymore. You can simulate those spectra to a very, very high degree of accuracy based on initial principles. We know the chemical sheets. We know the spin interactions. We know pretty much everything about them so that we can actually simulate the spectra to a very, very high level of accuracy. And these are the simulated spectra that you see here, and we sum them up. All we need to solve is how much of each metabolite we have. These are the coefficients, A1, A2, blah, 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 all the way to AN. So this is the aim, to find the coefficients of this linear set of equations. So this is essentially a linear Resonance type of problem. And this is why a program that I will show you in the next slide is called LC model, linear combination model. Okay? So once we define those coefficients, these are the coefficients that we're interested in because these are related to the metabolite concentrations. This is an example. This is an output or one of the outputs from one of the most popular quantification programs. It's a model published by Stephen Provencher in 1991, and ever since is the most popular now. It's open source. Now download it or take it on your Linux system or even on your Windows system. But still, a Linux system is preferred. So this is LC model. It, as an input, takes a basis set. This is exactly what I said before. This is the simulated data for each and every metabolite. And then it tries to play with it, stretch one up, stretch one down, move everything up and down, make it a little bit broader, make it a little bit narrower, fuzz it a little bit. Eventually, we get a fit. And the fit here is the red line. You can barely see the black line that is behind it, which is the experimental spectrum. But what we can see is the residuals, which are the differences between the red line and the black line. And you can see it's a decent residual line. It's not perfect. We still have some non-stochastic differences, which can be improved. This is actual data from one of my experiments from Leiden. This is a 7 Tesla data set. And just to kind of guide you through the output, first of all, we have the metabolite concentrations. How do we obtain the concentrations? We use a reference. What reference do we use? We can use the actual non-water suppressed signal from the same volume, which gives you the amount of water. If you know how to characterize that, that's very deeply, which I'm not going to dwell on now, but this is something to bear in mind. Repeat it also in the next slide. Or you can express it as a ratio to the total creatine. Why total creatine? It is a hypothesis. The hypothesis is the total creatine almost doesn't change in very well. We saw earlier a syndrome where there is no creatine at all, so there is a caveat to that. But overall, across ages, across even diseases, the amount of creatine, at least in standard applications, may be considered normal, kind of relatively stable. Not in tumors, things change dramatically. And then importantly, there is an estimation of the error in the quantification of each and every metabolite. And it is given by something that is akin to the coefficient of variation. So it's the percent of standard deviation from the actual value given. It is also essentially the lower limit on the standard deviation, or the Cramer-Rao lower bound, CRD. So we see the literature, CRD, we see coefficient of variation, mostly CRD. And at the bottom, we have two numbers that can help us estimate quality. As I said, we will not talk about that, we just mentioned it. One is the full impact maximum, or the line width. Again, 10 hertz at 7 Tesla is good. 5 hertz at 3 Tesla is good. It's given here in ppm. You can barely read what it says, but if it's 0.029 ppm or 0.03 ppm, at 7 Tesla, it's 3 hertz per ppm. Multiply those two, it's 10 hertz line. It's quite a good line. You can see that it's a high quality spectrum, and also an indication of the SNI. Now, with respect to scaling and calculating actual concentrations, one has to be very mindful of lots of kind of things that can kind of throw you away from actual kind of, let's say, responsible quantification of those metabolites. So something to consider. So, you know, choosing between total creatine and water, each and everyone has pros and cons. So proton density in tissue varies across tissue types. So if we choose our volume or water from the volume to be our reference to calculate actual concentrations in millimolar, we have to be mindful that concentration of water in CSF much higher than concentration of water in gray matter and different from that in white matter. So we have to estimate those. We can estimate them from an image, from a proton density image, or we can rely on the literature numbers that pretty much kind of give an estimate to those numbers. One thing to consider is that CSF doesn't contain metabolites at all. Almost lactic and glucose in CSF. The rest are intracellular metabolites. But that means, and this is why I gave this picture here from the beautiful study from Brooks et al. In cerebral cortex on spectroscopy in aging, is that if you look at the brain of a 27-year-old and a brain of a 68-year-old, chances are that the amount of CSF, for example, in the intrahemispheric fissure, increases. And that means that if you scale your signal to the water signal and you don't take it into account, that you just increase a lot of CSF, it doesn't have any metabolite in it, you have an artificial reduction in the concentration of all metabolites without having that happening actually. Concentration in tissue might not have changed at all. So the difference in amount of water in tissue between those can create spurious correlations. For example, if you have a healthy subject versus subject with some sort of an inflammatory disease, and let's say that there is an intracellular or extracellular edema in those subject, you again scale your things to something that does change between groups. So one has to be very careful about this. And most creatine can vary, okay? But the advantage of creatine that it's not present in CSF. That's one thing that to be said in favor. Okay? So metabolite concentration is also very different in gray matter. Right? Not very different, but the differences are big enough so that the differences in the amount of gray matter and white matter within your volume adds an explainable variance. Because we know that, for example, well, we could have seen actually on the spectrum before. Choline is much lower in gray matter than in white matter, really much lower, it's about 50%. NAA is slightly higher in gray matter than in white matter. So these are differences that we know, and we can incorporate them, of course, in a statistical model to regress them out. We have to do this based on segmentation of tissue within the volume in which we require the data. This is important to do. Take your T1, coincide it with your, first of all, do the tissue segmentation, then measure what is the fraction of each tissue type within the volume, and make sure that you regress it out when you do your measurement. Summary, to finish it all, we have talked about pre-processing, and we talked about quantification and the difficulties in it, and we said that it is crucial to perform strict quality control, and the estimation of tissue fractions also helps you in the quantification. Finish with this slide. We have a lot of recommendations. Again, we're obsessed with acronyms and recommendations. That's the spectroscopic community. One of the recommendations that I would recommend to read, or the two papers that I like most, are those that are in NMR and biomedicine. I will send you gladly references to them. And this one is about the minimum reporting standards. Again, kind of puts you in the frame of thinking of what people that will review it actually look at. If you measure something that has common lower bounds that are so big that no matter how many averages you took, it completely meaningless. If you correct for tissue fragments, what kind of consequences do you use? Are signals actually distinguishable from one another in the field in which you made this? A lot of things to consider and to report. And that concludes my tirade on spectroscopy for today. Thank you very, very much. And I have to run check my cat. So I really, really apologize that I am not even staying for questions. But please send me for come visit. There to assist everyone in those experiments. I hope you enjoyed it. And if you want to do, let's say, more advanced like Java editing and non-proton spectroscopy, things like this, just let me know. Let Chris know, and I will make another similar presentation with some more advanced. Thanks.
Thank you.
And apologies for this haste in parting away from you.
Thank you.
My pleasure.
Well, we won't be in here next week. We're in the Genome Center seminar room, and it will be Peter giving a talk about encoding models in metabolite. So
It's about mention Sussex as well.
Oh, yes, please don't. Well, you all get emails. We are having a party, and the party is called CISC Day. We're having a kind of an open day to show the capabilities of CISC, to encourage people to collaborate with CISC. December 8th, between 10 and 3. Registration starts at 9:30. It's going to be in the medical teaching building. Everyone is invited. There is enough room in the Michael Chowan Theater to have everyone. There will be a set of talks in the morning, and then breakout groups discussing potential collaborations within the academia, within the industry, and also with clinicians. So all are welcome. Lunch provided, and even drinks in the afternoon. I may be lying, but hey,